right. Looks like people are still trickling in, but we're live now. So welcome everyone to yet another session of Neurolaunchpad. We are on session 7, I want to say, 7 out of 10. So if you have, just before we start, if you have any questions, please use the Ask a Question box, which is down to the right of your screen. And if you want to hear one email from us per week, talks for that week, and get just a single email, we're not going to spam you with anything, click the little Google Doc form that's there to the right, and just it takes about 10 seconds, and you can sign up, and we'll just send you one single email telling you who's talking. It's usually going to be a trainee, postdoc, or grad student. That's the whole point of this series. Everyone's welcome to attend. Faculty especially are welcome to attend. But it's just we want to keep a focus on trainee neuroscience and kind of highlight the people who are actually doing the work and who need it the most at this point of their careers. So with that being said, I'm going to hand it off to our moderator and co-moderator for this session, Dr. Schaefer and Mengmi. So I will shut up now and hand it off. Thanks a lot for coming.
All right. First up, we have Amanda McQuade. So she's a senior PhD student in the Blurton Jones lab at UC Irvine. She has already published a first author paper and a review article and has several middle author papers. So super productive. And one paper looks like in Press and Nature Communications that I think we'll hear about today. All her work has been related to using human iPSC-derived microglia to study neurodegeneration. So the title of her talk today is Human TREM2 Knockout Microglia Fail to Activate Towards Alzheimer's Disease Pathology. Thank you, Amanda.
Thanks for that introduction. I also want to thank the organizers for putting this great seminar series together and inviting me to share some of my work. So, Alzheimer's disease is the most common form of progressive dementia. It affects half of people ages 85 and older. And the disease is identified by several pathologies: the buildup of beta amyloid protein plaques in the brain, tau neurofibrillary tangles, and these lead to a severe loss of the neurons and synapses in the brain. One of the problems to this date in developing therapies for Alzheimer's disease is the timeline. So the pathologies of beta amyloid and tau will begin to build up in the brain about 10 to 15 years before patients see declines in their memory and come into the clinic. So by the time that we're trying to actually treat these diseases, the brain has been degrading for almost a decade. So what we need to do is better understand these earlier stages of disease so that we can both find better biomarkers that will allow us to identify patients before the brain decline occurs and develop targeted therapies against the specific causes of these protein buildups. I've been doing this by studying the genetic causes of Alzheimer's disease. So large-scale genome-wide association studies have identified new cohorts of Alzheimer's disease risk genes. So these are the genes that are associated with increased or decreased risk of Alzheimer's disease across the entire genome. And you don't have to read these names, but I've boxed in red here the ones that are exclusively expressed or most highly expressed in the immune cells of the brain, called microglia. The problem now is that we don't know the individual function of these risk loci and how they're changing microglial activity. Microglia do many things in the brain. They're involved in synaptic pruning, especially during development. They will migrate to sites of injury, whether or not that's a plaque of amyloid beta or traumatic brain injury. They also have traditional monocyte macrophage-like immune responses, including phagocytosis of dead cells or proteins and cytokine secretion. So we need to figure out which of these microglial functions are changed by mutations in these different risk genes so that we know which functions we'll want to develop therapies towards. What's known so far about microglia in Alzheimer's disease is really the existence of this disease-associated microglia cluster, which was identified in 2017 by Ido Amit and Oleg Butovsky's groups, who showed when taking single-cell sequencing of microglia in a mouse model of Alzheimer's disease, at the beginning stages of disease, we had microglia not expressing activation markers since there's no pathology in the brain yet. But over the course of this disease development, these microglia will transition into what was named a disease-associated microglia state. And it's still being debated right now whether or not this activation is a consequence of the disease and it would be bad to have these activated microglia since they're occurring with disease, or whether they're actually occurring in response to disease and are helping to try and fight this pathology. So this is still being debated right now, but I'll show you some data today that I think we do need some of this activation of microglia. This data was all discovered originally in mouse models of Alzheimer's disease, but our lab has recently validated this as well with a human chimeric model. So we transplant human microglia into a mouse model of Alzheimer's disease and do the same single cell sequencing to look at the disease-associated microglia. We found that the human DAM signature is unique. There are about 350 genes that only turn up with the human cells in a mouse brain, but there is also a core of about 80 genes that are expressed both in the human disease-associated microglia and mouse cells. The gene that we're going to be talking about today is one of those genes, triggering receptor expressed on myeloid cells too. So it is an Alzheimer's disease risk gene, but it also is expressed in response to disease within this disease-associated microglia cluster. But first, I want to talk to you a little bit more about the model that we use. We've crossed the 5X FAD mouse, which is a familial model of Alzheimer's disease, to the Mittergy mouse. This is a mouse that has been humanized for 5 cytokines. Most importantly to this study, it has a humanized MCSF, which is the ligand to CSF1 receptor that is crucial for microglial survival. And so the mouse ligand will not bind the human receptor, so we need to use a humanized version of this ligand. When we cross these 2 mice together, we get what we're calling the 5X Mittergy mouse. This is an Alzheimer's disease model mouse that allows for long-term engraftment of human microglia. So we're transplanting actually our human precursors to microglia in postnatal day 1 or 2 mice so that they develop and differentiate into microglia naturally within the brain and are exposed to pathology as it is occurring in the brain. How this looks is shown here. So we get actually very good engraftment of our human microglia. In green, you're seeing a human-specific microglial marker, P2RY12, and our microglia in this case are mostly engrafting in the forebrain because that's where we've transplanted the cells, but we want to be studying the hippocampus and cortex, so this works really well. This model has been validated extensively in a paper published from our lab last year in Neuron. So I won't go into it too much today, but if you're curious more about that, you can read that paper. But today we're just going to be talking about triggering receptor expressed on myeloid cells 2, or TREM2. TREM2 is a membrane receptor on microglia, which has made it of great interest to pharma and biotech companies who are developing antibodies and small molecules that target TREM2, which then signals through its co-receptor DAP12 and has these different downstream functions. Loss of function of TREM2 is actually what increases AD risk. So we're going to be modeling that today with a CRISPR knockout of TREM2. That is going to be an increased risk of Alzheimer's disease line compared to the wild type. What's known so far is that TREM2 mutations, these loss of function mutations, actually lock our microglia in a homeostatic state. So what that looks like, this is again from our chimeric model where we're transplanting human microglia into an Alzheimer's disease brain, waiting 6 months for this pathology to build up, and then pulling out just the human cells to do single cell sequencing. When we do that, we get 5 different transcriptome clusters. The first is a homeostatic cluster, which expresses canonical microglial markers such as CX3CR1 and P2RY12. We really call this cluster sort of an unactivated cluster in that it doesn't express activation markers for microglia. We see our first activated microglia cluster, which we've called HLA. These are expressing a lot of antigen presentation genes. Of course, we also have this DAM cluster, which is marked by CD9 and TREM2. There's a smaller interferon high cluster and a very small degranulation cluster, which we're not really sure what the function of this one is yet. So this data was actually taken from transplanted human TREM2 or TREM2 knockout microglia. So if we look at just the DAM cluster, for example, and recluster this UMAP, recolor it with TREM2 wild types in gray and the TREM2 knockout microglia in blue, we can see that this DAM cluster is really mostly consisting of the TREM2 wild type cells. There aren't very many TREM2 knockout cells. Similarly, the HLA cluster is also enriched for wild type cells. I think this is a little bit easier to see in a pie graph. So when we have our wild type microglia that are within an Alzheimer's disease brain, about half of these cells still remain unactivated. And that makes sense because we're taking microglia from all over the brain, not just where the pathology is. We get a decent proportion of these microglia that turn on the antigen presentation genes, and about 10 to 15% which become DAM or disease-associated microglia. However, when we take a TREM2 knockout microglia from the same brain, about 3/4 of these cells are remaining unactivated despite disease, and a very small portion of them turn into the disease-associated microglia. So this is what I was mentioning before in terms of when we have an Alzheimer's disease risk microglia, they actually are not activating. So it seems like some activation will actually decrease your risk for Alzheimer's disease, at least in these early stages. But how does this look in the brain? So for this experiment, we co-transplanted wild type cells that express RFP and TREM2 knockout cells which express GFP into the same brain. You can see just by looking at the RFP and GFP signals alone that our wild type cells are activating and clustering around the amyloid beta plaques, which are shown in blue, but our TREM2 knockout cells do not. They remain tiled homeostatically across the brain. If you remove the amyloid from this picture, it would be hard to tell that it's even an Alzheimer's disease mouse. But we didn't just want to look at these cells, we also stained them for a DAM marker called CD9. So this is something that increases when the cells are exposed to disease. And showing the co-localization here, it's pretty clear to see that it's only the red wild type cells that turn on this disease marker CD9. The TREM2 knockout cells do not seem to be doing this. And we quantified that here. We also quantified localization to the plaques. So we showed that around the plaques, there are more wild type cells that have activated and migrated towards these plaques than TREM2 knockout cells. And that's the phenotype that I want to talk about today. Why are our TREM2 knockout cells unable to migrate towards plaques? With the idea that potentially if we could get these cells to activate closer to the plaques, we may be able to turn on some of this DAM activation phenotype and help these cells fight against disease. So to do this, we moved to an in vitro model of migration. And this is a two-chamber system. So we have an inner chamber connected by microfluidic channels to an outer chamber. In the inside chamber, we plated beta amyloid producing neurons and astrocytes. We decided to do this instead of just plating beta amyloid because in the brain around plaques, there is significant gliosis and neural dystrophy that occurs. And so these cells might be responsible for the signals that are calling the microglia to these plaques. On the outside, we plate our either TREM2 wild type or knockout microglia and see if they migrate towards this inner chamber. We were very pleased that this assay replicated what we see in vivo, which is that the microglia shown in white here, if they have functional TREM2, they're able to migrate into the center where the damaged and diseased neurons are, but the microglia lacking TREM2 do not make it into this inner chamber. Importantly, we also looked at microglia motility by doing a scratch assay, and both wild type and knockout cells close a scratch at the same time. So our knockout cells are able to move, they just don't perform directed chemotaxis. We did next RNA sequencing to try and figure out in an unbiased manner what receptor ligand pairs could be involved in this phenotype. And we found several receptor ligand pairs, but today I want to talk to you about CXCR4. So CXCR4 is a G protein coupled receptor that is activated by the ligand SDF1 alpha. This was interesting to us because SDF1 alpha is highly expressed in the hippocampus, which is where a lot of AD pathology begins. And there's some data showing that SDF1 alpha can increase microglial recruitment to plaques. We also found from our own data looking at the human disease-associated microglia signature that human dams increase their expression of CXCR4. And we hypothesized that it could be that they need to turn on CXCR4 to migrate towards these plaques and induce the rest of the signal. So by our RNA sequencing, we found that CXCR4 was deficient in TREM2 microglia, but we wanted to validate this at the protein level. So we did immunocytochemistry and flow cytometry, both of which showed that our TREM2 knockout microglia do express lower levels of the receptor CXCR4. We next wanted to show that this deficit is functional, so we did calcium imaging. CXCR4 is a G protein coupled receptor, so when the ligand is bound, there will be a calcium transit. We did single cell calcium imaging using ratiometric flu-4 and fluor-red dyes, where flu-4 will increase its fluorescence when it's bound to calcium, and fluor-red does the opposite. These images are from the peak signal, where we've just exposed the cells to SDF1 alpha, and the wild type cells have both a greater number of cells responding to SDF1 alpha than the knockouts, and on a per cell basis, they actually have a higher calcium signal. Here's the average data over all of the cells, and you can see the darker color with the wild type cells having a robust response to SDF1 alpha, but the TREM2 knockout cells do not. So far we've seen that our TREM2 microglia don't migrate properly. They have lower levels of CXCR4, and that is a functional deficit. But we still needed to link back our CXCR4 data to the actual migration phenotype. To do that, we used a small molecule AMD 3100, which is a CXCR4 antagonist, to see if this was sufficient to block migration to our pathology. Here's the data that I showed you before, where we have wild type cells migrating towards beta amyloid producing neurons and astrocytes, and knockout cells not migrating towards this pathology. Then we took just wild type cells and blocked CXCR4 specifically using our small molecule inhibitor, and we see that this was sufficient to decrease migration into the inner chamber. Interestingly, TREM2 knockout cells, when you compare them statistically to the wild type cells treated with just a CXCR4 inhibitor, there's no significant difference in the migration. So it could be accounting for the entire phenotype that we see here. In summary, what I've shown is that when you have human wild-type microglia and expose them to Alzheimer's pathology, they will turn on a unique disease-associated microglia transcriptome profile, which includes CD9, and they will migrate towards plaques or beta amyloid-producing neurons and astrocytes via CXCR4. In an Alzheimer's risk condition, which we modeled by knocking out TREM2, these cells do not activate towards the disease and cannot surround plaques. So this, more generally, could mean that it may be therapeutically beneficial to actually heighten microglial activation in patients with these TREM2 mutations, at least early in disease, to try and get them to respond to the pathology. In the future, what we're working on is studying CXCR4 function now back in vivo, where we've been overexpressing this in our TREM2 knockout microglia. We still need to answer the question of whether or not getting our microglia around the plaques is sufficient to rescue a dam phenotype, or do you need the dam phenotype first to migrate, sort of a chicken and the egg situation? We're investigating several other receptor ligand pairs as well, which we found from our single cell or from our bulk sequencing data, such as the purines and CCL2, CCL3. And I'm very interested as well in more broadly studying the function of CXCR4 in Alzheimer's disease. For example, I do study another Alzheimer's disease risk loci called MS4A6A. And when we did sequencing on this loci, which is actually protective, we found an increase in CXCR4. So what we talked about today is TREM2 loss of function, which increases AD risk and has lower CXCR4. But with MS4A6A, where decreased expression decreases risk of Alzheimer's disease, we find higher CXCR4. So it may be that many of these Alzheimer's disease risk genes are working through common pathways, such as CXCR4, which would be therapeutically beneficial. I want to thank my lab and everyone who has been involved in this project, specifically Yijun Kang and Amit Jerman from UCI and University of North Carolina, Charlotte. And I'm happy to take any questions, or if you don't feel like asking them here, you can follow me on Twitter or email me. Thank you.
All right. Thank you so much, Amanda. That was a great talk. Maybe I want to make sure there's no questions first. Before I definitely have questions, but I want to make sure there's questions for the audience get answered first. No? Okay. Okay. So, Amanda, I have a question about IL-34 in your model, because you use humanized CNSF, but there's, in the mouse brain at least, there seems to be IL-34 and CSF1 sensitive cell populations, or at least that's based on the secretion of the molecules. Do you think it's worth trying to look at how the transplantation works with IL-34?
Yeah, so as I understand it, the mouse IL-34 will bind the human CSF1 receptor, but we do know that if you have a mouse that has IL-34 normally, but not MCSF, the microglia will not survive for more than a couple weeks. So it does seem to be more reliant on MCSF. But in our in vitro model, we have both IL-34 and MCSF to ensure survival. So yeah, they both do signal to the same receptor.
And my other question was, with your CXCR4, so TREM2 also binds A beta. Do we know that TREM2 binding, so could it be that, do you think CXCR4 is also driving the gene signature changes, or do you think it's responsible for getting microglia to the right place, and maybe like binding A beta is what drives the gene signature?
Yeah, so it's sort of unclear. I mean, TREM2 has been proposed to bind to many, many different things. In the paper that was recently published in Nature Communications, that a lot of this work is from today, we show also that ApoE seems to be, if you don't have TREM2, our microglia will phagocytose no ApoE. So it suggests that potentially even TREM2 is the only relevant receptor for ApoE in these cells. But yeah, we're not sure whether or not migration is sufficient to induce a DAM phenotype, or whether or not it's just CXCR4 is part of the DAM phenotype that you get either from binding A beta originally through TREM2, or yeah, it's not clear yet.
Okay. Do we have some questions from the audience?
We don't have any questions right now. Megmi, feel free to interject.
Yeah, thanks, Amanda. I think that's a very wonderful presentation. I don't have a specific question to ask you right now, but I think probably we can pull questions from the audience, but I don't see any. Why don't we just move on? Then probably the audience needs some time to think about it, and then, yeah.
Sure, thank you.
Oh. I have a question, if you don't mind, but it's kind of silly. Is the actual function of microglia is the activation of microglia associated with the actual changes in the size of the plaques near?
Yeah, so microglia can compact plaques and make them sort of less fibril. So they usually have these long sort of fibrils off them, but they can compact them. But again, it's something that we're not really sure whether it's good that microglia are compacting plaques, because then there's less of the plaque to be touching neurons and killing neurons, or if it's bad. There's sort of also these ideals that if you compact plaques and these plaques are taken up by the microglia, that then they're changed somehow in the lysosome, so that when the microglia die, it could be even seeding more plaques. So I think there's still a lot to learn about what we want microglia to be doing and not doing at the different stages of Alzheimer's disease.
So there's no clear, is there no behavioral readout longitudinally of like whether it's good or bad to put in microglia in specific parts of the brain for the survival of the mouse long term, like survival curves?
So
and stuff
there
like that?
are some groups that have studied ablation of all microglia in these mouse models, and they showed that actually there were fewer plaques. But there are now newer papers showing that ablating microglia in different ways hasn't replicated that. So I think there's just a lot to learn about what these cells are doing and what the different ablation methods, how they're acting differently.
It was super clear. really interesting. Yeah, awesome.
Thank you.
Sorry, Megmi, we do have a question.
Oh yeah, I got that. So we have a question from Hao Chen. He's asking, have you tried overexpression of CR4 to rescue migration phenotype in your in vitro assay?
No, we haven't yet overexpressed. I think that is something that we're working on doing, but we're going back just because it's more relevant in vivo anyways than doing these experiments in a mouse to see if they actually overexpression is enough to make the microglia migrate. Good question, though.
Sounds good. I think we can move on then, right? Thanks a lot, Amanda. That was fantastic.
Thank you. All right. Next up, we have Erez Geron. He's a postdoc in the GAN lab at NYU since 2016. He did his PhD at the Weitzman, the Shiloh lab. He has a number of papers currently in review exploring dendritic plasticity during sleep and learning and memory. And the title of his talk today is Retrieval Spikes, a dendritic mechanism for retrieval-dependent memory consolidation.
Yes, that's true. Thank you. So let me share my screen, and indeed, I'm going to talk about. By the way, you hear me, right? Everything is okay. Great.
Yeah, everything is good.
Yes, that's wonderful. So
You just need
let's open it. So I'm indeed from Wenbiao Gan's lab from NYU, and it's very nice to be here. Thank you for having me. And one of the things that interests Wenbiao and many other people is how memory forms. And it's well established that when memory form, they form through a series of stages. First, you hear something, you encounter something, you encode in information. Then this information is being consolidated. It's being stabilized. It's being more resistant to interference. And after that, usually people think about that in the context of sleep. After that, the memory is stored in our brain. It can be there for our entire lifetime, and until one day you have to retrieve that information. And hopefully, if you don't have any significant problems, you can find that information, bring it back to mind, and use that information. So one of the things that mediates consolidation is retrieval shortly after encoding. And for example, you can think about coming back from a class, looking at your notes. Let's say your handwriting is not that clear. It's a little bit of acute retrieval of something that is not clear, and that is known to induce strong consolidation, like a very good good way to improve memory retention. And that's what we're going to talk about today, how retrieval shortly after encoding induce consolidation, and by that improves memory. So so that's like the basic finding that was known for more than 100 years and is basically the second strongest effect on memory that is known. And all this work is done together with Bayan. She's great, and she's not here, so I just want to acknowledge. So how can we study retrieval-dependent consolidation in mice? Like everybody else, use the classical conditioning paradigm, in which you basically pair a tone and a shock. The tone is a bit long, the shock is a bit painful, and then a day after, you present just the tone to the animal, and you see if the animal freezes. If it freezes, then it's associated the tone with the shock, and consider it as a predictive measurement, a predictive cue for pain, and that's why it's freezing. So this is how it looks like. Now I'm going to show you how a mouse that was trained the day before behaves in a chamber that is new to it, and once it's hearing the tone, it's not moving, it's freezing, even after the tone is gone. Let me just show you the part with the tone. See, the time runs, and the animal just reorients towards the speaker, And once the tone is passed or like not heard anymore, the animal continue to explore this chamber. So the readout is very easy. So what we wanted to do to study retrieval during training or early retrieval was basically to do this, add the tone immediately. Oops. Yes. Add the tone shortly after pairing and see how it affects freezing the day after. And we saw that if you just take one pairing, you get about 50% freezing. And if you add just the retrieval tone unenforced, non-enforced, you get much more freezing, almost or as you would get if you would condition the animal with two pairings. We did another kind of set of experiments in which we trained animal under the microscopes. Here, these are animals that are head restrained. This animal underwent surgery, so the freezing levels are are actually quite significantly lower, but the basic phenomena is the same. Adding a retrieval cue during training again improves retention and again improves memory. So the question is, okay, you've got all that, what are you going to do with it? So we decided to study the motor cortex. Motor cortex is very associated with behavior, and we had some experience in our lab. We know that silencing the cortex, the motor cortex, during training interferes with acquisition of conditioned freezing. And we also know that it's leading to reduced activity of the motor cortex. And the general idea is that if an animal wants to freeze, it's better have less activity in areas that promote movement. That is, you want to freeze, let's have less activity in the motor cortex in response to the conditioned stimulus. So that's what we did, and we just wanted to see that the motor cortex is not only important for a conditioned freezing, but it's all for the acquisition, but it's also required for retrieval-dependent consolidation. And what we did was basically inject animals with ARCH-T. ARCH-T is an optogenetic silencer. Control animals were injected with the same locations with GFP. And if you'll take a look at this part of the screen, if we just take animals, condition them with one pairing and then give them light, the light doesn't matter much because these are animals that express GFP, then you get some kind of freezing. If you give the tone and light, you get more freezing the day after. But, and this is the critical experiment, if you inhibit the motor cortex the day during just retrieval, just the second tone, they don't learn as well. They actually learn as if they didn't hear it. And if you give the tone at the light at 10 minutes delay, you don't get interference, meaning that we basically didn't kill the motor cortex, we just interfered with retrieval. So let's continue. We basically trained the animal under the microscope to look at their neurons, and with the specific locations and the specific cell type of neurons that we're studying are pyramidal neurons, layer 5, which are the predominant output cells of the cortex. So to conclude this part, we've got a phenomena that we want to study, which is retrieval-dependent consolidation. We've got a paradigm, we give them the other tone and we see what's going on. And we've got a relevant cortical region, which is the motor cortex, and a relevant cell type, which is the output cells of the cortex, pyramidal neurons, layer 5, or also known as layer 5 pyramidal neurons. So we're looking at these neurons. We're imaging while we're shocking them and while we're playing the tone again, and we're doing that with two kinds of preparation. One are transgenic animals, and the other one are animals that were infected as neonates. And to begin with, we're looking at their dendrites, apical dendrites in layer 1, a very integrative area of top-down inputs. And let me show you one video. What's happening is that we're playing the tone, giving the shock. Here's the shock. Some spikes are appearing. And then we will play the tone again in a minute, and much more activity in those layer 5 pyramidal neurons. And this phenomena also occurs in single labeled neurons. We give the tone, we give the pairing, nothing much happens sometimes. And then we play the tone again, a big spike, and that was basically the basic observation that we had. And we can quantify it. For example, this is the quantification of this animal. And if we can quantify a lot of animals, then we see that once we give the cued recall, the reminder, what sometimes we call, then you get an increase in the tone. And I'm not showing you, but I'm telling you that if you don't give the reminder, you don't get this kind of elevation. If you train the animals with an unpaired conditioning, you don't get this increase in dendritic spiking. So it seems to be like a real phenomena. So we call these spikes retrieval spikes, as they are induced by retrieval. And we see a couple of phenomena that increases the certainty that they are related to learning. One is that freezing correlates with retrieval spikes. The more spikes you see, the greater the prevalence of spikes within a population in the motor cortex, the higher the chances the day later the animal would be a good learner. Retrieval spikes are transient. They occur only within the first 15-20 minutes of training. If you release the animal and return it after 15 minutes, you would still see it. But after that, after 30 minutes or 120 minutes, the chances to see a reduction in activity is growing. So it's some kind, it's not the thing that the motor cortex wants to do in the long run. We think that it's actually a learning process that directs the change. And in line with that, we see that like retrieval spikes correlates negatively with later responses to the tone. We see that both at the level of the soma and with the dendrite, and both the dendrite with the dendrite itself. If we're looking at dendritic spiking during training or 1 hour later, and we've got this data for also 8 hours later, basically you get a negative correlation. The higher the response to the tone, the greater the decrease later on, negative correlation. So spikes, okay, a lot of spikes. What do spikes do? Spikes basically do two things, especially in the apical dendrite. One is that it's a good way to communicate with the dendrite, the apical tuft, with the soma, right? There are hundreds of microns away, sometimes 700 or 800 microns it might be. So a huge burst of calcium is a way to affect the soma and drive it. And the other thing that they do, they induce plasticity, induce and coordinate plasticity. So those spines that are active just before and during, and they are very active, they experience such large amounts of calcium, for sure they're going to change a bit, right? So now that we know this, let's take a look at two measures of post-synaptic, and with the what I told you 2 slides ago about how the spikes are transient, we can also understand the videos better. So for example, here we've got what we call a high resolution imaging of the dendrite. These are sibling branches and a parent. And 2 videos, one is after 1 minute after pairing, and one is 1 hour after pairing. The animal was released and returned to the home cage. And here, once the reminder will start, you will see that there are many spikes, like this. Let me just stop it. And here, once we play the tone, it's again a reminder, but we keep the reminder for early times. Here it comes. No spiking. Okay, and what we thought was that probably there is some kind of learning rule that is being applied here, or some depotentiation of spines, and that's probably a lot of things that are happening, but that's what we wanted to study. So we measure 2 measures of postsynaptic activity or postsynaptic plasticity. One is the change of spine activity. You can see that we can easily measure the spine activity over time, and the other thing is to measure the spine size, changes in spine size. Both things we can do pretty easily. So if we're looking just at the overall postsynaptic input, the overall postsynaptic input is reduced. And I know that it might be also presynaptic, but we'll deal with that soon. And if we're looking, okay, what's going on with the postsynaptic reduction, we can see that it's mainly in the spines that are most active. If I'm taking spines from each animal, I'm dividing it into quadrants according to activity, and I'm looking how did each quadrant changed. The most active spines are those that reduce their activity. If we look at size, if I'm taking, here we've got about 200 spines. If I'm looking at the relationship between size change and activity during training, and I can take different measurements, such as total activity during retrieval, total activity in general, during activity during spikes, during strongest spikes, and whatever, everything that is related to retrieval gives us more or less this significant reduction according to activity. So the more active you are, the more reduction you will have, or more chances you will have to be reduced. If we're dividing it by animals, so here I'm dividing each animal to each spines from each animal to quadrants. In actually all the animals we've got here, I'm showing 7, but we've got, or 8, but in other conditions, we've got similar results. All the time, the most active spines during retrieval, during the largest spikes, are being reduced in activity. So I'll try to shorten my talk, but one of the nice things that people are always talking about in plasticity is how the timing of activity affects plasticity. So we took a look at that. Usually people talk about presynaptic and postsynaptic cells. In the case of dendritic spikes, they're talking about spines or inputs to the dendrites and the firing of the dendrite, not the cell. And we took a look, for example, at this example. So you see that we've got here a number of spines that are active. Basically, the spike starts here, but let's say it's 300 milliseconds apart. All those spines that I flagged here were depotentiated, while the majority of the spines actually potentiate. So that's a strong, a nice example. If I'm taking all my data and I'm checking what's the chance or what's the involvement of spines to be active before the 5 strongest spikes, if they've got a low involvement before in the second and a half before those spikes, they will potentiate. If they are very involved, they will depotentiate. Again, some kind of anti-Hebbian plasticity, which means that once you're involved, you get depotentiated to reduce the response of the cell to the tone, to the conditioned stimulus, to enable freezing. And maybe the last slice with data is that dendrites are known to have different compartments, different branches that carry different calculations, and we can take a look at that. For example, here you can see those 2 branches that I was showing you before. One is colored in red, the other one is gray, and if we look at these 2 branches, we see that they've got different properties. First, the spines on the gray one are much more active. The x-axis is the activity of the spines during spiking, and the y is the change in activity. Second, the gray branch, almost all of the spines, except for one that was very inactive, depotentiate. On the other hand, this one shows a mixed population, where the ones that are not active potentiate, and ones that are more active depotentiate. So, and another thing that we found is that the branches that show predominant depotentiation are the branches themselves are more active, their spines are more active, and also the spines are showing earlier activity before spiking. But the nice take-home message from this branch-specific plasticity is that it seems that they've got different rules. While here, the governing rule is if you're an active spine, depotentiate. Here, the governing rule is if you're on this branch, you will depotentiate no matter what, because the branch is so active. And I think this is the first indication of branch-specific plasticity rules, and we really like it a lot. So just to show you one last thing, because I showed you basically until now just correlations, so we used something to inhibit activity after spiking. So we took something by Ryochi Yasuda. He took CaMKII inhibitor. CaMKII is the master regulator of plasticity involved in LTP, but also in LTD. We can block it by introducing this into cells. We can do it very sparsely and shine blue light immediately after we see the spike. So the condition would be that we pair the animal, pair the tone and the shock, then present the reminder and give inhibition of CaMKII immediately after the retrieval spikes. And as a control, we give the CaMKII inhibition immediately 5 minutes after. And then return the animal and do the reading afterwards. So in the control animals, we get basically the regular readout, but if we inhibit calcium activity just after the spikes, we don't get normal depotentiation, and also we lose the reduction in dendritic activity. And I can also tell you that if we do that in densely labeled animals, we don't get learning. The animal don't learn, they don't freeze, while these animals, do learn and do freeze. So to conclude, we wanted to study how does retrieval improves memory. We did that in mice. We trained animal under the microscope. We discovered that retrieval evokes calcium spikes in the apical dendrites of layer 5 pyramidal neurons. And this retrieval induces a weakening of synapses and branches that are very responsive to the tone. And I say that the last point is F. So thank you. I'll take questions. After I'll take Bai again. She's really great. And Wenbiao, which is also very, very great. And all the lab members of Wenbiao of the lab. And if you've got questions, I'll be happy to answer them.
Thanks so much, Erez. So I think we already have a couple questions.
Sure.
Shall I
Maybe
pause?
yeah, go ahead.
Okay, sure. So the first question we have from Susan. And so the question is related to the saliency of the task. So would you think your observed spine depotentiation is related to aversiveness of the foot shock? And another question is, would an attractive memory lead to the potentiation of the involved spine in motor CX or elsewhere?
What's the second question again?
Okay, so would an attractive memory lead to potentiation of involved spines in motor CX or elsewhere?
So I'll answer the first one, and I'm not so sure about the second one. So no, it's not. The potentiation and depotentiation are not related to just getting shocked. If we just shock the animal, if we give unpaired, which is shock, tone, and then we play the tone again, we basically get a very different plasticity and very different responses to the tone. So just shock doesn't explain it. It's actually the retrieval. The retrieval tunes the learning into the tone. Yeah.
Just to clarify, I think what Suzanne is at is like the shock is a bad thing. I think this is what the way I'm interpreting it. The shock is a bad thing, and you're looking at depotentiation as an important readout. Therefore, if instead of a shock as the unconditioned stimulus, you were using something positive, like something appetitive, would you get instead potentiation as opposed to depotentiation?
No. The answer is no. The question is, the idea is that we're looking in the motor cortex, and if you want to move less, then you will have depotentiation and elimination, and if you want to move more, you will probably have a potentiation. The Luti lab had a paper, a Nature paper, obviously, in which they basically train the animal to jump when they get the shock. I would argue, or I would think that in this paradigm, they will have a fear, and they will have spine potentiation and formation because the animal learns to move more.
Cool, thanks. And we have another question from Susan as well. She's asking, so you are talking about dendritic caseal spikes, but you are only imaging the apical dendrites. And how can you be sure that the effect is not due to the backpropagating potential?
So we did all these experiments also in the stoma, and we also imaged the interneurons that target either the stoma or the dendrites. So we're not very concerned that that we're missing something very big here, at least. And in general, backpropagation in layer 5 doesn't really reach the distal area. It's not a big concern, but I can tell you that we don't see this elevation in the stoma of pyramidal neurons. The activity at the stoma is basically high after the shock, but it doesn't increase after the reminder is presented. So we really think that the purpose or what these spikes serve is plasticity. It doesn't drive the cell. We see a lot of inhibition onto the stoma and very little inhibition onto the dendrites. If we image NDNF cells and if we image SST cells. So inhibition to the dendrites is actually pretty low during reminders.
Thanks. Yeah, I also have a question like from my own. So I'm curious a little bit like on the part where you talk about like different branches of the spines, and one is dominant in potentiation, the other is not. Like, what are the governing rules, or like what are the physical properties, or maybe I don't know, like how does it evolve to be different?
Yeah, that's a question that is easy to ask, but is hard to answer. But it is a good question. Yeah, basically, I think about it in a way of let's say there is an LTD threshold, a dendritic LTD threshold, that once enough calcium is being released, something comes out of the dendrite and gets into the spine. But that's like purely. Yeah, there are probably like a large number of things that govern. We actually try to branch-specific plasticity is something that was demonstrated in a number of tasks within those neurons, and yeah, we're definitely trying to figure it out. But yeah, actually, this is a progress.
Maybe there's another question in the chat, I think.
Oh. Yeah, sorry, I missed this one. I think it's the question from Mary. So she's asking, is there a motor response with the freezing behavior that is being reflected in M1 as opposed to a consolidation event?
So once an animal is a good learner and you present the tone, you would see a reduction in activity both at the dendrites and at the stoma of the pyramidal neuron. So basically, and this is something that was actually also shown in human and rats, that your motor cortex, is even a paper called your motor cortex is freezing when you freeze, something like that. But during this consolidation, or what we're showing, we actually see an increase in activity, especially in the apical part. Yeah, this is the association layer. So it makes more sense that some changes will be done. Maybe there are changes also next to the stoma. We can't image it so well.
And what is your future direction that you're gonna like study or develop a little bit more?
Yeah, publish a nature and open a lab. There are tons of things that we can do here. There are tons of things. Because basically people always studied preconditioning before and after. And if they did that, and even those that bought those fancy microscopes, they can only do it at the cell resolution. And basically our simple apparatus, which we describe in detail, with the manuscript, is basically very easy, and you can get, as you can see, spine data, and it doesn't cost you a thing once you've got a 2-photon microscope. So there are tons of things that you can do. And yeah, I think that you can do it in Alzheimer, right? We know exactly which spines change. So you can do it in a model and see which spines change and which spines don't change as the disease progresses. Yeah, there are tons of things.
Have you?
I had a quick question. Sorry, go ahead, Maggie.
I want to ask, like, have you looked into the quantal content or quantal size when this like plasticity like changes, or like, how does it affect learning in general?
Under which condition? What manipulation?
Yeah.
I don't understand which manipulation.
So so you have this like retrieval immediately after the condition. So I'm just wondering how does the quantal size or quantal content change with respect to this different like retrieval immediately after or long before?
Talking about post-synaptic release and stuff like that.
Yeah, that's right.
Yeah, so we basically studying the post-synaptic, as I heard one professor said, it's about 1 million times easier, but we can basically refer to spines as the detection unit of presynaptic release and normalize it. Mm-hmm. We see that the strongest, so if we take like all the spines that we got and we image it, we normalize it according to themselves, we see that the most spine activity occurs after the shock or shortly after the shock during the spiking. And after that, it's the retrieval. But during the shock, there's also a lot of inhibition. So there's usually less spiking immediately after the shock because of that inhibition. So the system is really poised to allow retrieval to spike.
I had a question, like, I was wondering, like, what you think, because I think you had a plot, right, where it's like if you do the retrieval tone, 15 minutes is good, right? You'll get a big response, and then as you start titering that time a little bit longer and longer, that kind of starts disappearing, right?
Yes.
So, what do you think? I guess if I were to just like anthropomorphize super quickly, I'd be like, oh, okay, does that mean that I need to revisit my notes in this extremely short amount of time after the initial event? What is that time window, do you think? And why is it in the minutes time scale? What could do that? Is it a neuromodulator tone? Is it something else that's doing it? How do you think that happens?
Yeah, great. I wouldn't conclude any general conclusion into life from that. But it does help to. But if you want, you can look on the testing effect. That's something that got a lot of attention in the last 10 years and got much more relevance to learning the testing effect. So regarding that thing, so there is a basically comparable phenomena that if animals take mice, condition them, and then administer extinction sessions, the animal don't extinguish their fear. It's called early extinction deficits. It's like something that is a little bit known, but basically people try to see why there is such a time-limited period in which extinction can't happen. It's very similar to what we show, only we show that it's a little bit, in our case, it's a little bit shorter. So there is different modulation of arousal during that time. There is basically about an hour and a half in which, after you get a shock, in which adrenergic signaling is very, very high. And the other, so one part of the answer is arousal. Arousal goes down. And another thing is that once you give retrieval after a period of time, the memory is already different. If you don't give the reminder, and you look how the plasticity of the spines change, you see that they already go down. They already depotentiate. But plasticity is much more wide. It's not related to the spines that were necessarily active during the tone at the pairing. So the substrate of the memory, once it's retrieved, is different if it's 2 minutes after training, after pairing, or if it's 1 hour after pairing. So I would say two sides to this.
Got it. Yeah, that's interesting because I just don't think of, neuromodulatory tone, or I mean, arousal makes sense, right? Like maybe you keep like some level of adrenergic signal stays up throughout like this whole time. It would be nice to use some of the new optical sensors or something to just monitor that in real time and see if there's any obvious candidate for plasticity that stays up in the order of many minutes, right? Because I definitely don't think of cholinergic tone or adrenergic tone or even dopaminergic tone of varying in that time scale for a task like this. But that's an interesting answer.
Yeah, it doesn't have to be. Yeah, well, we've got just like a couple of minutes. People show that like adrenergic activity after shock is high for at least 1 hour.
Oh, gotcha. Okay, cool. Yeah, I didn't know that. Yeah. All right. Do we have anything else?
I guess that's about it from the audience.
Great. Well, if there's no other questions, I think we'll wrap it up. We're at an hour. So about it. Thank you very much, Dr. Schaefer, Megmi. And
Thank you, everyone.
Great talk, guys.
Yeah.
Thank you so much.
Bye-bye.
Nice meeting you. Yeah, bye.
Awesome talks. See you next time.
See you at the SFN.
Thanks a lot.